This next presentation covers drug development and how we move forward from where we are through in that process. So yeah, over to you when you're ready.
This is going to be very similar to what I talked about last year. So if you were online last year, you can get a glass of water for 30 minutes or something. And but let me and this would be a good sort of talk to interrupt me and ask questions. So please interrupt, ask questions. Happy to do it. So again, disclosures. So I think for those of you who are new to rare disease and drug development, we want to think about what are all the stages that it takes to make a therapeutic. It seems really simple, but it's not. Mostly because of we're not allowed to do things in people that we're allowed to do in mice for good reasons. And so there's a lot of regulation and a lot of need to do it carefully and slowly. So one thing that we pound away on in our clinic and in when you're developing disorders is describing the condition. So you can't, and this is why we're going through patient collections and what we call phenotyping, or really trying to understand the nature of the condition. Because if you're developing a therapeutic, you need to be able to say, well, this is going to get better if I give the therapeutic. So things like, is arterial hypoplasia, like I was showing you before, is that something that we should go after? We see it in different territories, the aorta pretty much, and as we've been quantifying in most territories. What about other things like fibrosis of like skin fibrosis? We have some features that we've seen in patients that have exuberant fibrotic. This is just an example because sometimes it can happen on the inside of the body, not on the outside. This is just easier to see. And so is that something that we can see and try to modify? Or what about things like pericardial effusions, where we know this happens at a higher rate in people with Myhre syndrome, where this fluid collects, around the heart. Is that a good thing to target? Because, maybe if we had a medicine, we could see whether this happens less if people are on the medicine. But you can't actually propose to do that until you know how often this actually happens in the first place, because you don't know if you're going to help if you can't see it and how it happens. So what you have to have is what we call a natural history study. And that is what's happening to people with the condition under current care. So it doesn't have to be with people who aren't taking any medicine. That's actually wrong. It's people who are taking the medicines that are commonly used for the condition right now, and we want to try a new medicine on top of that. So we want to understand the natural history of patients with Myhre, even if they're taking losartan, or even if they're taking a different blood pressure medicine or different meds. And we need to understand it over time and how it changes over the lifespan, so that we can target a therapeutic range. You can't do a 20-year clinical trial. That doesn't help folks. It takes too long. So you have to do things over a short period of time. And so you have to focus on things that change over a short period of time. And because of this, we need to work collaboratively as groups, because, these individual reports are good. I think it helps us understand things, but for a natural history trial, we need more people and more data. We have to understand to a degree how the disease works. So we need to understand SMAD4 and how this particular gene change affects SMAD4 function. The reason for that is when you're making a medicine, you have to understand if you're hitting the target or not. it's a requirement for the FDA is understanding the on-target effect of your medicine, if it's a new medicine. And how, you know, the gene change affects the cell, and then later, say an animal model, and then what you would predict it's going to affect a person. But we're ahead because we know a lot about SMAD4. I was showing you this slide earlier, but we know we already have assays for these things that have been created over time in biology. You know, that many of us in this field have been running for years. So I think that puts us ahead. What about things like cardiac fibrosis? This is something we want to study, but we couldn't get to this from just routine clinical care. We'd have to do extra scans. You know, we'd have to do more detailed MRIs, for instance. So we can't, we're not going to be able to do a kind of natural history study. We're going to need to go beyond just sort of clinical observations of what we do regularly in clinic. We're probably going to need to do additional testing and really understand what's going on. Similarly, would we want to target high blood pressure? Well, there's lots of high blood pressure medicines already, right? So we're going to use those, and we do use those in patients with Myhre syndrome. But you know, is that something we want to go after clinically for a new medicine? Everyone's. And we need to understand how SMAD4 drives those conditions, and what are the interactions of SMAD4 with the other SMADs, and how the dysfunctional molecule is affecting the biology of the cell, and what are its adjacent binding partners and interactors that mediate the mutation and the way the mutation expresses itself in the disease. So those are things that need to be identified. You know, Hong Mei, who I mentioned previously, is working on this in collaboration with Dr. Lin and myself, looking for these types of clues. Something that the animal can also help us with. And then when you think about therapeutics, there's lots of different ways. There's repurposing drugs, so drugs that already exist. That would be an example in this community, would be something like losartan, where it's already an FDA-approved medicine. I certainly, you know, in the face of high blood pressure, think it's a very reasonable choice, and has good sort of scientific backing, if not clinical evidence, at least a good rationale for its use. But then we also want to develop newer therapeutics, whether they be, you know, small molecules, or whether they be something more advanced. So the things you need for therapeutic development involve picking first, which part of the disease are we going to treat? You know, you want to treat everything, but you can't run a clinical trial where you test everything. So you need to pick some of the manifestations, you know, whether they're going to be cardiovascular or lung or neurologic, and decide that that's what we're going to do a lot of heavy phenotyping in, and then evaluate how our medicine works. Target identification is an important part in on targeting of that therapeutic. So what is the molecule going to be something that goes in and targets SMAD4 directly, and therefore we have to prove that it's modulating SMAD4 function, or is it going to be modulating something downstream from SMAD4, kind of like an effect from the SMAD4 mutation, and it's going to be on target to something downstream that could still be beneficial from the standpoint of the disease? But you have to figure that out, especially for what we call a new drug, or an investigational new drug, and that's what the FDA wants you to prove. Obviously, you have to pick the modality. It's going to be a small molecule, or you're going to go after something like gene therapy. And then you have to have a model, especially for rare disease, to test it in a preclinical context. And then finally, the how do you know it's working? The only way to do it is to do a clinical trial. So disease manifestations, this is a multi-system disorder, and what are we going to go after? I think cardiopulmonary disease is a good thing to go after because we have lots of technology to evaluate it. And so we're more likely to see subtle changes, subtle improvements. If no medicine is perfect, no medicine is an easy cure. So you need to understand small improvements as well as large. And then what about, neurologic disease seems to me to be super important because it can have the biggest chance for improved quality of life. So that's something I want to focus on. Target identification, I think we went over this before, but like, how do you decide what these non-SMAD4 targets are? You have to go through a process of experimentation and looking in the known literature, testing it out in the mouse, and figuring out what is either, are we going to go for on target for SMAD4 or a related molecule? And then picking the therapeutic modality. There are ways now, and this is something we really want to do, is to target the SMAD4 gene directly through genomic engineering. This is becoming a reality in some disorders. It's easier to, our task is a little harder here because in some conditions, you can just delete the gene. So that's a little easier to delete a gene. It's always easier to destroy rather than build, but it's true in biology too. Like for this amyloidosis, you can just destroy the amyloid gene and it's helpful. There's approaches like this being tried for cholesterol and actually working, where some of the genes that produce cholesterol in the liver are now being targeted and are going to be pretty effective for atherosclerosis. I think the data is going to be presented in November, the first part of the data, the American Heart. But could we do it for SMAD4? And I think that's something that we want to try in the animal models. The hard part is, how do you get it there? We have these enzymes that can reprogram genes and change the sequence, but the hard part is to get it into the cells, and how do we ensure that is safe? So these are things that we're all working on for multiple disorders, not just Myhre syndrome. And then how do we test it? I mean, the good news is that we have our mouse, and the folks in France, have a mouse. And so I think there's going to be more than one shot on goal about trying to do this. And the other good thing is, as I would ask for good news in the last one, is that we have phenotypes. so the mouse causes disease, and so therefore we can test whether fixing it helps the disease. It seems rather simplistic, but it's a requirement. And then how do we, if we have a medicine, how do we test it? The general way we test medicines is through what we call a randomized clinical trial, and it's certainly the best way because at the end of it, there's no questions about did it work or not work, because you're literally randomizing. So there's no variables, there's no outstanding variables that aren't controlled for. But you have to have a lot of people to do randomized trials, and Myhre syndrome and other ultra rare disorders are not conditions that we can randomize. So we have to do things like basically pre and post treatment, where we get a patient, we study them very well, we understand their physiology for several years, and then we give them a medicine, and then see did it work by comparing how they were doing after the medicine to how they were doing before the medicine. And that's what a lot of what we need to do in rare rare disease. And that's what helps. That's what you absolutely have to have a good natural history trial. The other thing you have to do is have a lot of money. And we're working on it. We're trying to go from stage 1 to stage 2 here, in our group, and I'm sure in others. And so we're trying to, stage 1 is to get some government funding. We may be able to leap ahead if we have an effective gene therapy that we could try to move to a bigger resource allocation. But clinical trials are super expensive, and they cost in the 8 digits for sure. So that's part of the challenge, and part of the thing that foundations help with is to grow that interest. So that's why I put these slides in, and happy to say them again just to reiterate to the patient community. But what can the patient community do with the goal of having a therapy is to help researchers like us or others, whoever has good ideas, get attention from our government and from European governments and from biotech and pharma companies. It helps to participate in natural history studies, whether that's a survey or a more detailed natural history study where you're coming into a medical center and having some MRIs, and that's what will help us interest these other entities in investing more, because they can see that we have very clear idea of what the disease process is. Because the FDA doesn't, if you have to have very clear-eyed goals and very straightforward, measurable, what we call biomarkers that accurately reflect improvement of either disease or quality of life in patients. That's what they're looking for. Having an animal model with preclinical data, we're working on that. And then the other way is, to steer the priorities. So what the patients say is the most important thing to them is what the FDA is responsive to. In fact, when we do what's called an IND or a investigational new drug meeting with the FDA, we often bring along patient representatives to speak to the FDA and say, this is part of our goal as well. So just in conclusion, it requires a lot of planning and clear goals of therapy. I think Myhre syndrome being genetically pretty straightforward is helpful in terms of trying to find therapies. And I think patient groups, including this foundation, can really positively influence this. And again, thanks a lot.
Thank you, Mark. That's amazing. We've got loads of questions, so I'm glad we've got a little bit of time to dive into those. First up, you mentioned attention from pharma, and just interested on that point. Do we get interest from pharma as a rare disease organization as small as ours, excuse me, just by proving something within our community? Or do we get attention from pharma because our results can be a model for something larger that could help other heart diseases, etc.? So what's your view on that?
I think both, for sure. So, and I think I may have touched on this in the past, but I think that's the right way, is both that there's a rare disease. There are rare disease gene therapeutics that are now in clinical use. That is something that is engaging to pharma. However, you have to have a lot more evidence than we do right now. But from a foundation standpoint, getting out and meeting with people and just getting the word out that this condition exists and that it needs a therapy is super helpful. because you're educating people who are already in the rare disease space but may not know about Myhre syndrome, so they can sort of put it on their radar. And then the other thing that you alluded to is important. Like if while we're really in depth studying the pathophysiology of Myhre syndrome, if that pathophysiology relates to more common disorders, which I think it does, and one of my goals in the research is to tie those things together, then that becomes extremely interesting to a pharma company, because they can see that, well, there's a limited number of patients, and frankly, then a limited number of dollars that some drug could make. If it relates to a bigger patient population, then it expands that reach. That has to be, though, that's usually like a small molecule more than like a gene therapeutic.
Okay. Great, thank you. A question here: can you target fibrosis generally, or does it have to be system specific? So could a treatment help muscles or joints and the heart?
Yes, absolutely. So one of the ideas behind using drugs like losartan is that you not only treat blood pressure, which losartan does by antagonizing angiotensin 2, is that it has a side effect of antagonizing fibrosis. That was noted many years ago in the kidney literature, and that was the rationale actually for losartan being used in conditions like Marfan syndrome and Loeys-Dietz syndrome that affect the TGF-beta signaling pathway. And similarly, it's the rationale for folks who are using it in Myhre syndrome. And I certainly use it so far in patients who have hypertension, but we may build a rationale for using it even earlier if we see beneficial effects in the animal model. Some physicians, and I think there's nothing wrong with this, are using it prior to cardiovascular symptoms. And in a rare disorder like this, I think that's totally reasonable. We need to. The only reason it's reasonable is because this is such a safe medicine. It's
Okay.
very. It has such a good safety profile that the downside is pretty low.
And that actually nicely feeds into another question on here. So if people are already on losartan in the community,
Mm-hmm.
which we know they are, how do we track this already if they are taking it? Or how does that data become more formalized? Can it?
Well, so you can do, what we call a retrospective study, where you go back and you ask people their symptoms prior to losartan and their symptoms after. But that kind of data is, on the scale of data, quality is not the highest. And I don't think people should. I think if your doctor recommends to take losartan, then take it. But it does make it difficult to quantify that data and to know whether it's really helping.
And
Yeah,
again,
it's very hard to get around that particular.
People are asking here, what benefits have you and other providers seen with losartan in patients?
So Angela or some of the other folks may be able to speak more broadly. I understand the group in Italy thinks that it's helping, joint symptomatology and so forth. I'm not in a position to opinionate about that. It's not something I do.
Sure.
But I mean, from a cardiovascular standpoint, it lowers blood pressure. That's what it's supposed to do.
Mark, among our MGH patients, we are not tracking that because we don't have patients that we've personally prescribed it for. But I would say Nicola in Naples is still very interested in this. I don't think he's on this conference right now, but that is still his interest.
Yep.
Great.
And
Thank you,
I would just say, you know, I deal with a lot of rare disease groups, and I think if you're taking a medicine and you feel better on it, like, that's okay. Like, you don't need to worry about the science. Like it's makes you feel better. That is good.
If somebody wanted to take this or were taking it and they had a doctor that was monitoring, you don't want to totally do it yourself. You could use the paper that Nicola wrote. The first author is Capuccio. And he lays out in his methods, which is a standard thing in a paper, what they did. For example, to talk about joint stiffness, you can't just say you're feeling better. There's actually a way to measure the range of motion. There's a scoring system for firmness of the skin, the Rodnan score. It takes a lot of training. So it's kind of up to you. If you want to add to the science of what's going on, there is a way of doing it. We're not trying to deny people who want to feel better, but this is how we're going to decide if it is useful for everybody.
Lovely. Thank you, Angela.
And just a caveat though, Mark, by the way, Mark, just amazing talks. He mentioned that losartan is very well tolerated. It's easily available. Half the adults on this conference are maybe using it, but there is a time where you do have to be careful about it, and that is in the setting of possible kidney disease. So you have to sort of be looking at the whole big picture. Does that make sense?
Yeah, and I'll add to what Angela just said by saying we still use it in the presence of kidney disease, but we want to have a nephrologist on board for that and monitoring kidney function. If somebody already has kidney disease and we're thinking about using one of these medicines, then we want to have one of those physicians on board.
Great. Thank you for clarifying that. We've got quite a specific, we have lots of amazing people on this call. We have community members, we have researchers, scientists. So let's dive into some of the detail. In the mouse model, do you see increased SMAD4 protein levels similar to the human? SMAD4 PROTAC degraders might be one potential drug modality.
Yeah.
Mark, do you want to first translate that question for us?
Sure. So in when you say in humans, I would say you have to caveat that by saying in a couple of fibroblast cell lines from humans in one study, which was a good study, but limited just by the nature of the way these things work. We don't see that. SMAD4 in the animal looks like it's about the same. So what they're referring to, one of the hypotheses from the original description of Myhre syndrome was they got fibroblasts, skin cells, more or less, from patients, and then skin cells from people without Myhre syndrome. And they stained for SMAD4 protein, and there was more SMAD4 protein in the disease, suggesting that maybe there was either increased production of SMAD4 or decreased degradation. And the authors, hypothesized that there was decreased degradation of SMAD4. It's a totally reasonable hypothesis, and but in the animal model so far, we don't see that. So I think I haven't seen good human data on whether or not, I mean, like human meaning actually like literally biopsies from people showing that SMAD4 is upregulated. And at least in our animal model, we can get the disease without more SMAD4. Now, PROTAC, what the individual there was speaking to is a technology that can go into cells and degrade a particular protein. And for sure, if you could degrade the mutated form of SMAD4 selectively with some technology, the PROTAC or other, you could probably improve the Myhre the Myhre syndrome conditions, although loss of function of SMAD4 causes a different disorder. And so we need to, it's hard, this is going to be a balance. It may be better to have loss of function than gain of function. So it may end up at the end that's okay. You just have to take it, and maybe that works out okay. But you don't really know until you test. So I hope that answers whoever asked that.
Great. Thank you. Question here about the natural history study. Can you take retrospective data? So can we use previous medical records?
Absolutely. So the way a lot of natural history studies work is that patients are enrolled. We reach backwards and get all the pre-existing medical documentation that they have, and then separately do some prospective testing, like regular imaging testing, or for brain stuff like regular MRIs and advanced technologies, basically. So the answer is yes. When we design a natural history study, there's a retrospective component and a prospective component.
Mark, do you mind if I jump in for a second?
Please.
I just want to clarify what you were thinking of, and just so the audience hears too. Currently, what we are doing at MGH is the patients who come to us, our main goal is we want to help you and your family. Number two, everybody that we see is part of sort of a general, non-identifiable collective data pool. And this is the data that we're currently working on for our big paper. It is not designed specifically as a natural history study. It's designed, it was a prospective study, and we're looking going forward. Several patients have come back more than once. In addition, though, there is a future dream for a true natural history study, which would extend beyond MGH. I just want to clarify what Mark was saying is that we do not accept medical records from people that we have not seen. It's just a matter of resources and the ability to really process them. And, would you
and that's
talking about?
Yeah, I would say that's true right now. But if we're able to get funding to do a prospective natural history trial, then we'd be able to enroll people who enroll from wherever. But yeah, I think what Angela said, like currently, what we're working with is what we can do.
And something Andrew and I have talked about a number of times, numerous times over the years, is this ambition for a natural history study. But just to give context, and what Angela was saying as well, is obviously MIRI affects every body system. So when we're looking at medical records, it involves a huge amount of data, a huge amount of information, and that all obviously takes a large amount of resource. So as Mark mentioned, that comes with a cost attributed to it, but there's definitely ambition from all sides for us to get that moving.
And by the way, you've seen me and Mark, and behind the scenes is the really fantastic clinical research coordinator, Eleanor Simone. And we want to thank the Myra Sooner Foundation for supporting her salary. Thank you.
Thanks, Angela. Right, okay, we're going to go much wider here with a kind of a big picture question. AI, that's had a lot of chats around drug development, discovery, machine learning. What's your thoughts, Mark, on how that impacts the research and drug development process?
I think, yeah, so it's a really excellent question. So the applications of AI in drug discovery there's different ways that it can be applied. Basically, it's a way to do AI is just pattern recognition by a computer program. It can recognize patterns that humans either sort of pass over or are so minor that we don't think a lot of them. And so sometimes they can pull out patterns and information that humans don't. That's basically how it works. So when you think about all the different stages that I went through there about drug discovery, you can apply something like AI to different stages. So one thing that has been done for AI is to put it on the beginning side, like to do human phenotyping. Basically, are there things in the body in using medical imaging that if a computer was looking at that could see a pattern that we don't see, like So we've applied that to aortic disease and aneurysmal conditions, and we're able to expand the world of aortic genes by a lot doing that kind of AI. In small populations, but that was done across the UK Biobank with 50,000 MRI scans. So you're starting with a lot more information there. In Myhre syndrome, that we could attempt things like that, and I think if we had anyway, it would be more difficult. The other way AI can be used is in the drug development space, like literally in the time at which you have a target like SMAD4 and you're trying to understand molecules that bind the SMAD4, and you're trying to like shift through them and get a category that can, that you wouldn't recognize otherwise might bind your molecule. So those kind of things are done with AI, but have been done for some time. It's not as new as I think you would read in the lay literature. But yeah, that's the other common way of using AI. I think the things that I read about that are kind of fanciful are things like understanding outcomes from using drugs using AI, and that's not how medicine is done. That's a venture capital. That's like a pitch deck at a conference. It's not reality. In reality, when we use drugs, we need to like see what they do in people, and it's unfortunate, but that's the reality.
Sure. Thank you. Question here: how do you see the landscape changing for drug development and discovery with organizations like the Chan Zuckerberg Initiative and Every Cure trying to bring together a wider collective of knowledge? Is collaboration like this going to help the smaller rare groups like Myhre?
Yeah, I hope so. I mean, I think the Chan Zuckerberg Initiative is great. I mean, the more rare disease research, the better, because these other groups are dealing with similar challenges. So similar challenges of small patient groups and individualized pathologies that need to be unfurled. And so, I mean, I think that's part of that. I'm glad that those groups are catalyzing that. We're trying to do that same thing. We have a rare pediatric vascular disease study group here at MGH that I'm keep putting Myhre syndrome in front of them. But it's those economies of scale, because every single rare disease can't reinvent the wheel. So I think yes, I think it's great. We should try to get Myhre syndrome in front of those people.
Yeah, great. And I know this is like a difficult question for you to answer, so we won't hold you to it, even though we're recording it. But how far away are we from getting treatments for Myhre? And I guess this is in the context of that journey, that slide. I love that slide where you said we're on that path to then getting NIH government funding. Like, how far are we from that part to that part? Because obviously we need to attract the funding that doesn't come from just the patient population, which has done such an incredible job to get us to the place we are now. Like, what do you feel like is the journey time for attracting those NIH grants?
Oh, well, we're writing them now. So I hope we'll be moving from stage 1 to stage 2, in our group. And the life cycle of NIH grants is difficult because a lot of times take years to acquire, and Congress doesn't help, because every time we don't pass a budget, everything gets delayed. It's federal funding and is affected by the federal government, and so I think we're trying to transition from stage 1 to stage 2 now. I think we have good data, and we need more, but I think we're on our way. And then, you know, we're also trying to find funding for that natural history study that I'm talking about. And we're working on getting funding for that too, whether it's through Jan Zuckerberg or the NIH.
Excellent. That's, I think, a really amazing positive note to end on, that we are close and that you know, the ambition is there. And thank you so much for talking us through both of those presentations this morning. We know you're in high demand because I think you're off to speak at another conference this afternoon. Just to say to everyone, to remind you, if there's any questions you have for Mark that you think of after the presentations, do let us know. We will capture them all and we'll do our best to answer those afterwards. So thank you, Dr. Mark. That was really fantastic.
Thanks, Ed.